All right. So my name is Dante Bolzan. I'm a UC San Diego PhD student in the iLab, the La Jolla Institute for Immunology. And today I'm going to be talking about a deep learning model we developed called SCOPRE that performs sub-kilobase compartment prediction using a long short-term memory model. So because Hi-C and contact maps were already introduced today, I think I will just briefly remind people that a contact map is simply a matrix that tabulates the frequency of pairwise interactions between genomic loci across the genome. And so these contact maps, reveal the hierarchical organization of chromatin, and the largest scale feature that people typically identify using these contact maps are what's called AB compartments. And you can think of these as two classes that we can assign loci to. And A compartments are correspond to open chromatin, and B compartment corresponds to closed chromatin. And the thing to keep in mind is that loci preferentially interact with other loci that are belonging to the same compartment. And so how are these AB compartments computed? So given the raw contact map, you normalize the matrix to account for distance, and you get this Pearson correlation matrix in the end that essentially highlights this plaid pattern. And using this Pearson correlation matrix, we can apply PCA and obtain eigenvectors. Usually we use the first or second eigenvector. And using that eigenvector, we can then binarize it to label loci as either A or B. And so the way we do that is typically positive values in the eigenvector correspond to A compartments, and negative values correspond to B compartments. So the first AB compartments that were identified historically were done at 1 megabase pair resolution. But now with how sequencing costs are going down and how Hi-C technology is being further developed, AB compartments are being called at higher and higher resolution. And just last year, compartments were called at 500 base pair resolution. So this is sub-kilobase pair resolution in the lymphoblastoid cell line by the Jordan-Rowley group and ENCODE. And this was unprecedentedly high resolution for which they were called that. And they were able to do this by pooling 33 billion read pairs from various experiments. And using this massive amount of data, they were then able to call AB compartments as sub-KB resolution using this algorithm called POSSUM, which essentially is able to compute the first and second eigenvector of a very sparse and large matrix. And so why do we care about these biologically? Well, as you can see in the two figures in the bottom, these sub-KB compartments can be found in coarser compartments of different labels. And so as you can see in the bottom right here, for example, you can have a sea of inactive chromatin, and you can have these tiny transcriptionally active regions that contain loci that are far away in linear space, but are localized here. And so being able to call these sub-KB compartments, we're able to sort of understand the fractal nature of how chromatin is organized in the nucleus. And so AB compartments are cell type specific. And so the question we wanted to ask was whether we can call them in other cell lines other than LCLs. And we tried out multiple cell lines shown here, those with very deep sequencing data, and we were only able to call them in this fibroblast cell line called HFFC6. And incidentally, the number of intrachromosomal read pairs for this cell line is above the 7 billion read pair threshold that the authors of the POSSUM algorithm found to be a requirement to be able to even call these sub-KB compartments. And so given the high cost of Hi-C experiments and the importance of being able to call AB compartments in multiple cell lines, we thought about a way of calling them in the absence of Hi-C data. And so this is just outlining how we called sub-KB compartments in the fibroblast cell line. And so, it's just a standard procedure of pooling our read pairs, obtaining the contact map, and then calling our AB compartments, which we were able to confidently do in 17 autosomal chromosomes. And so going back to our problem formulation, we want to call these sub-KV compartments in the absence of high C data. And to do so, we developed this model called SCOPRE. And the way it works is that it takes 9 epigenomic and genomic features, which I show here. And so we bin the genome into 500 base pair intervals, and we take the mean ChIP-seq signal for these 6 histone modifications, as well as the mean attack-seq signal, which measures chromatin accessibility, GC content, and gene overlap. And so we feed these 9 features into what's called a long short-term memory model. This is a variant of recurrent neural networks, and these are a class of deep learning models applied to sequential data. And given the sequential nature of genomic data, I thought this would be apt for predicting sub-KV compartments. And I just want to highlight here that our LSTM module is bidirectional and stacked. And so the bidirectionality allows the model to learn information upstream and downstream of each bin. And the stacked nature, so these blocks here are being fed into multiple layers. So here there are 4 layers, and this allows the model to learn long-range interactions as well as more complex interactions between different bins. And so in the final layer, we concatenate the output and feed it into a fully connected layer with a sigmoid activation function, and we obtain a probability prediction for each 500 base pair bin, which we can then binarize to obtain the AB compartment labels. And so just to clarify, this model is trained on binary compartment labels. And if you're wondering what's going on in these LSTM cells, for the sake of time, I'll just mention that essentially they are computation that's from a set of weights that we learn, and it determines how much information we keep from the previous bin and how much information we'll add. And so it's a way for the model to learn both short-range and long-range interactions. And so in order to evaluate our model, we decided to first test within cell lines, because we need to do it before we try to predict in a cross cell line fashion. And so we employed a leave 1 chromosome out strategy, train on all chromosomes except for 1, and then test on the remaining 1. And because there are no other deep learning models that I know of that predict sub-KV compartments using these features, we simply ran 3 baseline models, non-recurrent baseline models, a support vector machine, logistic regression model, and a random forest on each of the folds. And as you can see in this bar plot, for each chromosome that was left out, SCOPRE achieves a higher accuracy than these baseline models, suggesting that recurrence is indeed important for predicting these sub-KV compartments. And what we found is that in most chromosomes, we are able to achieve 90% accuracy or higher. And we see the same story in the other cell line for which we were able to call these sub-KV compartments. And we achieve even higher accuracy, with an average of 94% across all chromosomes. And so here we are measuring accuracy, but we wanted to learn why the model makes mistakes. It does. And so one way to evaluate that is we can check if misclassified bins have low confidence eigenvector values. Because as you recall, AB compartments, while they are binary labels, they are derived from a continuously valued eigenvector. And we indeed found this to be the case. So this is the LCL cell line. And you can see that the correct predictions follow this bimodal distribution of eigenvector values, which is what we typically see. And the incorrect predictions are enriched around 0, suggesting that the model is perhaps sensitive to compartment strength. We also see the same pattern in the fibroblast cell line. So another way to evaluate the model is to look at the predicted probabilities, because at the end of the day, SCOPRE is predicting probabilities. And as these bar plots show, we found that the predicted probabilities are indeed higher for the correct classifications for both A and B. And we see this in LCL as well as in the fibroblast cell line. And so the next thing we wanted to do is see how well the model captures these small and large scale compartment intervals. Compartment interval is simply a region of consecutive bins that have the same compartment label. And as you can see here, we are capturing these large-scale structures. The first track corresponds to the ground truth possum compartment labels for this particular locus in chromosome 14, and this is in the fibroblast cell line. And the second track corresponds to our binary predictions. The third track corresponds to the confidence. And so not only do we capture these large-scale features, we also capture small-scale compartment intervals, like you can see here with these tiny A compartment interval. And even when the model fails to predict these binary labels, you can see a dip in the confidence level right here on the left, showing that the model is indeed sensitive, right, to these compartment switching. Next, we wanted to look at how how the feature profiles of correct and incorrect predictions compare, right, across different features for our model. And so what you see here in this heat map is that looking at just H3K27 acetylation, which is an active chromatin mark, you can see that it's positively enriched for the correct A predictions, which is makes sense, and negatively associated with, or not as enriched in correct B. And we see the same phenomenon in the fibroblast cell line. Now, we found an interesting case with H3K27Me3, in which we actually found positive enrichment for misclassifications in both cell lines. And we think this is because this chromatin, or this histone modification, has both active and repressive activity in cells. And so I have some additional results I don't have time to go over, but I will just briefly mention. We found that SCOPRE outperforms naive baselines constructed by thresholding features. We also tried to predict in a cross-cell type fashion, and we achieved a mean accuracy of 76% by using the trained models on LCL to predict on human fibroblasts. So it's still a work in progress to try to predict across cell types. And in terms of future directions, one way that we can generate more labeled data in other cell lines is to try to enhance the resolution of contact maps at 500 base per resolution, and then obtain an eigenvector. So that would give us more labeled data to do cross-cell type validation. And then finally, we could develop a completely different model that is able to incorporate a low resolution contact map plus these 1D features, and then try to predict these sub-KB compartments. And so I just want to thank my PI, Dr. Faradai, as well as everyone else in the iLab, and I'll take any questions. Thank you for listening.
We do have time for a question.
Yes, great talk. So I picked up on the fact that you didn't use the lower resolution compartments as a feature. I saw on the last slide that you're thinking more along the lines of upscaling the high C and then calling compartments. Right. But why not just use the low res high?
That is a great point. Actually, that is something I'm thinking of doing because it's a very easy thing to add to the model, right? Just create additional feature vectors, and you could even do it at multiple resolutions. Yeah. So I think that's a great suggestion. Yeah, it'll be interesting to test. Yeah, thanks.
So a quick question. If I followed correctly, you are, for instance, a classification problem. Is that right? Predicting A or B compartment?
Yeah.
Do you have any sense of whether trying to predict the eigenvalues would change anything in the formulation? Would that be noisy or helpful?
It's a good point. So with this model architecture, you can definitely do that. One thing I probably failed to mention, but we actually did try to correlate the confidence scores, right, that the model outputs with the raw eigenvector values. And we indeed find a pretty high correlation here, 0.9. So that is showing that even just training on those binary labels, we're able to, the model is sensitive to the compartment strength. But yeah, in terms of framing it as a regression problem, I think one issue is scale, right? Because across different chromosomes, you'll have a different, scale, right, for the eigenvector values. So you could do some sort of min-max normalization, and then, that would control for that. But yeah, it's a good suggestion. I think definitely it's something worth trying. Yeah.
Let's thank Dante again. Thank you.